Hey guys, so today in our AS biology lecture we are going to cover another important organelle, which is our Golgi apparatus. So the very first thing which you have to know, I've already labeled. So Golgi apparatus, this entire thing is actually called the Golgi apparatus. It's also sometimes called the Golgi bodies or the Golgi complex. So this entire structure is actually called the Golgi apparatus, Golgi body, or the Golgi complex. And basically this entire structure can be said to form a stack. So these are like sack-like structure which are like stacked together. Okay, so we can see there are many Golgi, these things, this sack-like structure, and there are many of them, so they appear to be a stack. So therefore we call it to be a stack of flattened sacs. Okay, and these sacs are actually called the cisternae. Okay, so each of these sacs can be referred to as the cisternae. Okay, so basically what happens is that there are certain incoming transport vesicles. So these one I'm marking as blue. These are the incoming transport vesicles which are transported from the rough endoplasmic reticulum. I've covered about the rough endoplasmic reticulum before video in one of the previous videos, so you guys can check it out. So the rough endoplasmic reticulum, as we know, has ribosomes on its surface, and these ribosomes actually synthesizes different types of proteins. And ribosomes, the main function is protein synthesis. So it transports these proteins to the help of these incoming transport vesicles. So the main function of this Golgi this Golgi apparatus or the Golgi body is actually the modification, or we can also say the processing of different types of molecules which actually gets carried to the Golgi apparatus. Okay, so modification and processing of different types of molecules, mainly proteins, which actually come from the rough endoplasmic reticulum. It also modifies lipids. Okay, so these actually modification of lipids and proteins. So how do they modify? For example, they can actually sugars can be added from the Golgi vesicle, from the Golgi apparatus. Sugars can be added to proteins to form glycoproteins, and sugar can also be added to the lipids in the Golgi apparatus to form the glycolipids. Okay, so these glycoproteins and these glycolipids, they are actually used up for the synthesis of the cell membrane. Okay, so when we are going to study about the phospholipid bilayer in the later chapters, we're actually going to see how this actually works. So these glycolipids and glycoproteins are used for the synthesis of cell membrane or the phospholipid bilayer. Okay, also another function of the Golgi apparatus is actually these ones are actually are actually called the secretory vesicles or the Golgi vesicles. They're sometimes referred to as Golgi vesicles or secretory vesicles. So the main function, these secretory vesicles are actually butts off into secretory vesicles at these end. This Golgi body actually butts up into smaller vesicles which are round structure. These vesicles can actually form lysosomes. They can contain different types of enzymes. So these vesicles can either be secreted out of the cell through the cell membrane by the process of exocytosis, or they can be actually used for the synthesis of lysosomes. Okay, so what are lysosomes? We are going to cover lysosomes in detail in another video, but lysosomes are basically organelles which actually contain different type of hydrolytic enzymes which are used for phagocytosis and many other processes inside the cell. So these secretory vesicles or the Golgi vesicles can actually enter the lysosomes or used for the synthesis of lysosomes, and these Golgi vesicles can also be transported out of the cell out of the cell membrane by the process of exocytosis. Okay, and since we learned about the modification of proteins and lipids, these proteins, glycolipids and glycoproteins, can also help in the synthesis of the cell membrane or the phospholipid bilayer, which is another name for the cell membrane. Okay, the other function of the Golgi apparatus, as I was mentioning before, can be during plant cell division. For example, these Golgi enzymes. So there are certain types of enzymes called the Golgi enzymes, which are produced by the Golgi apparatus. These Golgi enzymes can be used for actually the synthesis of new cell walls. Okay, synthesis of cell walls. So we know plant cells have cell walls, so these Golgi enzymes can be used for the synthesis of cell walls. And also in the gaseous exchange system, in our gaseous exchange system, these Golgi apparatus can actually is used for the synthesis of mucin. Okay, or you can also say mucin or mucin. So this mucin is one of the main components for the synthesis or for the production of mucus. So mucus, you already know from O levels, is a sticky fluid-like structure. Okay, so these are secreted by the goblet cells in the lining of the trachea. So you learned about those. So yeah, so that's basically the content we have to understand for Golgi apparatus. But we are also going to talk about details about the structure when we later talk about protein synthesis in the later chapters in the central dogma of biology. So yeah, so once again, for a quick recap, this is called the Golgi apparatus. This entire thing can be called the Golgi apparatus, Golgi body, or the Golgi complex, and these are flattened sac-like structure which are all stacked up together, and they receive certain types of incoming transport vesicles from the rough endoplasmic reticulum, which contains different types of proteins secreted by the ribosomes on the surface of the rough endoplasmic reticulum, and these actually enter the Golgi apparatus, where they are actually modified and processed, and different types of proteins are actually modified to form glycoproteins, and lipids can be modified to form glycolipids, which are used for the synthesis of the phospholipid bilayer or the cell membrane. And we also learned that it butts off at this end, at the bottom end of the Golgi apparatus, to form secretory vesicles or Golgi vesicles, and these Golgi vesicles can actually be carried out of the cell through the cell membrane. This process is called secretion or exocytosis. Secretion or exocytosis. Okay, which is the release of these molecules from the cell membrane. So yeah, they can be used for exocytosis, and this entire pathway is actually called the secretory pathway. Okay, so yeah, and then we talked about certain functions of the Golgi apparatus. So they can also be used for the synthesis of ribosomes, and we also learned that during plant cell division, Golgi apparatus can be used for the synthesis or production of cell walls, and during gaseous exchange they use for the synthesis of mucin, which is a major component for mucus. So yeah, that's it. That's kind of the content which we need to cover for our syllabus level so far. Okay, so yeah, that's it for Golgi apparatus. So stay tuned for the next video, and thanks for watching.